So good morning, everyone. Thank you for attending Talita, Botti, Braz thesis defense. The committee is composed by myself and Dr. Talita, who was the co-advisors of the work. Professor Aurora Grapmates from the chemistry department of PUC-Rio. Professor Paulo Roberto de Souza Mendes from the mechanical engineering department of PUC-Rio also. Professor Ronnie Thompson from the Mechanical Engineering Department of UFRJ, and Professor Kerry Dutcher from the University of Minnesota. So Talita, I first thank all the committee members for their sharing their time to participate in taking part of this committee. And then Talita, you will give your presentation that will be followed by questions from the committee.
Thank you, Marcio. Hello, everyone. Thank you for the presence here today. I will present my work about the effect of interfacial rheology on the stability of water in oil emulsions. Let me see. Not working. Yeah. Emulsions are very common to be found in our day. Basically, they are a mixture of two immiscible liquids that, in the presence of an agent emulsified, can be stable for long periods of time. From the different applications, our work is motivated by the challenges found in the oil industry, where the emulsions are formed during all the production process, since the beginning of the exploration, because into the reservoir there are oil, water, and gas, and increases especially when we have the water injection to maintain the pressure of the reservoir and continue with the production. However, at the end of this process, it's necessary to separate this phase because successful separation is crucial to avoid problems during the refining process and maintain the high value of the crude oil. In this process, we can find both oil in water or water in oil emulsions, but our focus in this work will be in the water in oil emulsions. These emulsions tend to be very stable due to the presence of natural components such as asphaltenes, resins, and naphthenic acids, which can form rigidity in interfacial films, difficulty in the separation process. The destabilization of emulsions can occur of different ways, but the focus in this work will be in the coalescence process to provide the phase separation. Coalescence consists in a merge of two drops, and when we have the approximation of these drops, the liquid between them will be squeezed out into the bulk region, resulting in a convective flux of the surfactant in this sublayer. Therefore, we can observe that as the surfactant increases in this direction, it creates a weak zone in the middle of those drops that will favor the coalescence process. This is a complex process because it involves multiple flux occurring at the same time. Coalescence can be studied through different points of view. We can use a macro scale analysis when we evaluate the stability by the phase separation over a period of time, or using a micro scale analysis of a single event. This one can be done by using a droplet against a planar interface, or the coalescence between two droplets. And this last one can be done by using a microfluidic device that will allow to evaluate drops with small size. Before to present our work, it's important to see what it has been studied at the literature, and we can find a lot of works, but the focus here, it will be in which consider the presence of surfactant. In 1992, also showed that the film drainage rate is strongly dependent on the surfactant absorption kinetics, its interfacial activity, solubility, and interfacial rheological properties. Some of these properties will be evaluated in our work. And as a progress on the studies, we have several studies that show that the droplet residence time becomes longer in the presence of the surfactant because the Marangoni force that slow down the drainage of the film formed between this interface. More recently, Bochner studied the stability of emulsion by measuring the coalescence time as a function of aging time and asphaltene concentration. And they evaluated the coalescence of a drop against a planar interface. And our experimental setup will be an adaptation from them. However, we are going to use the coalescence between two droplets. They observed that the oil in water emulsions are less stable than water in oil emulsions, and they attributed this difference to the arrangement of asphaltene molecules at the interface. Another work that I will present it here is from Narayan, and they performed the experiments using the same surfactant used by us, and they saw that they used a microfluidic device to evaluate the coalescence of two droplets, and they observed that varying the surfactant concentration, the film drainage time is increases with an increase in the surfactant concentration and with the droplet size. So with this brief literature review and with our motivation, the objective of this work will be understand the separation process of water in oil emulsions with complex interface. To do this, we are going to characterize the water-oil interface. Then we are going to evaluate the coalescence between two water droplets. Then we are going to investigate in detail the structure formed at this interface. And finally, we will propose a new method to promote the emulsion destabilization. The idea with all this process will be provide the knowledge necessary for optimizing the separation process. About the materials used here and the rheological properties of this interface, we know that the crude oil forming complex, the crude oil emulsions forming complex structures at the droplets interface, and due to experimental limitations with the use of crude oil, we will use a model system that will mimic this complex behavior. So the materials used was a milky water as the water phase, and a combination with Primo, a mineral oil, and Span 8 as the surfactant. That is a non-ionic surfactant. And this combination will provide the complex interface observed when we have crude oil system. To obtain the interfacial rheological properties, we use a dependent drop method in which we have a drop suspended from the needle in the bulk liquid phase. The shape of the drop results from the balance between the interfacial tension and gravity, and the interfacial tension is obtained from the shadow image of this initial drop. By oscillating the volume of this drop, its surface will expand and contract sinusoidally, generating a stress at the surface that leads to the measurement of elastic and viscous dilatation of moduli. The elastic modulus is associated with the elastic response of the structured interface, and the viscous modulus is associated to a gradual diffusion exchange of surfactant molecules between the interface and the bulk region. So starting the results, we are going to present the evaluation of the interfacial tension with time for different surfactant concentrations. And we can see that the time evolution is strongly dependent on the concentration, the surfactant concentration. The critical micelle concentration was obtained using the interfacial tension at the equilibrium as the surfactant concentration, and is equal to 0.05% in weight. So analyzing these graphs, we can see that for concentrations lower and close to the CMC, the interfacial tension rapidly falls and then slowly approaches the equilibrium value. However, for the highest surfactant concentration, the surfactant diffusion to the interface, the rate of the surfactant diffusion to the interface is initially very fast, and we can observe that the interfacial tension decreases until reaching a first equilibrium plateau. And after that, we can see in this graph a second decrease until a new lower equilibrium value is reached. For this case, it was observed that this second drop on the interfacial tension occurs when we have formation of a solid-like film at the structure. We observed that reducing the volume of the droplet, and with this reduced, we can see some wrinkles at the interface that is characteristic of a solid film under compression. And this time was corresponded with the second drop decrease on the interfacial tension. So all analysis done from here, we consider the surfactant concentration above the CMC to provide this interface stability desired. Here we have the time evolution of the interfacial tension again, and the elastic and viscous moduli of the interface as a function of the surfactant concentration. We could define 3 different regimes as a function of the change in the elastic modulus. In the first one, we have the elastic and viscous moduli with values almost equal and both lower than the interfacial tension, suggesting that the interfacial stress is close to isotropic. And this initial plateau is attributed to the adsorption of the surfactant monomers or inverse micelles. And this region is correspondent to the first equilibrium plateau of the interfacial tension. In the second region, the elastic modulus starts to increase and becomes higher than the viscous modulus. And finally, in the third region, the elastic modulus reached the maximum value and then slightly falls at the same time that the viscous modulus decreases, reaching a value close to 0 at a long time. This region is corresponded to the second drop of the interfacial tension, as you can see in these graphs. For some conclusions with these results, the formation of a solid-like interface was identified by the increase in the elastic modulus, and we could see some wrinkles that can affect the measurement. So at this point, we are not able to affirm that these data are corrected once the dilatation of viscoelastic properties are dependent on the correct evaluation of the interfacial stress during these oscillations. Then this drop on the elastic modulus can be resulted of a measurement artifact. Once we characterize this interface, we are going to evaluate how they will influence on the coalescence of the droplets. So firstly, I will show the experiment that we performed to see the coalescence between two droplets. Here we have experimental setup composed by syringe pump, a pressure transducer, a high-speed system, and a cuvette where the coalescence are occurring. And this graph is corresponded to the pressure data used to track the events and obtained with accuracy the drainage time. Here we have some videos to better understand this process. In this first one, we have the droplets formation. So we started with the injection of the first drop. Then we moved down the cuvette, moving the lift. And then we injected the second droplet. So after the formation, we left them at rest in a desired aging time. Then we move these drops against each other. When the contact occur, we are going to measure the time to drainage this film between them, and finally the coalescence will occur. However, as we can see in this image here, for some conditions, the coalescence will not occur. Here we have the relation between the drainage time as the aging time, and we observe that two different behaviors. One when the coalescence will occur, and in this case the drainage does not show significant dependence with the aging time, neither with the surfactant concentrations, and the other one when the coalescence did not occur. Those results are extrapolated on this graph as infinite drainage time only to representation. And this phenomena was observed when we had the solid-like interface at this droplets. Correlating these results with the interfacial rheological properties, we can remember that when we defined the three different regimes to evaluate the elastic modulus, they were separated by two specific times. The T1, that is the time that the elastic modulus increases and becomes higher than the viscous modulus, and the T2, that is the time that the elastic modulus takes to reach the maximum value. Now we are introduce the TC, that is the critical non-coalescence time, and we can observe in this graph that TC is always higher than T2. It means that the non-coalescence requires more aging time than the time determined by the interfacial dilatational rheology. Now we are going to do a brief analysis of the behavior of the motions produced with this phase. And in this case, the emotions were prepared by a two-step method, starting with a slow addition of the water phase into the oil under constant steering with a magnetic bar. And after that, the emotions were steered by another two hooks. For the characterization, we used a turbiscan that measured the turbidity of the samples. This equipment consists of an infrared light source and two sensors that act simultaneously. Thus, the transmission detector will receive information from the light transmitted through the sample and is effective for transparent areas. And the backscattering detector will measure the reflected light and is effective for opaque samples. Here we have the results from this analysis, and we evaluated emulsions prepared with surfactant concentration below and above the CMC to see the difference. We have here a picture of these emulsions taken one day after emulsification, and the turbidity profiles of these conditions. And in these profiles, the blue zones on the bottom corresponds to the signal measured in transmission for transparent areas, and the orange corresponds to the backscattering for the opaque areas of the samples. The appearance of this blue zone on the bottom is characteristic of coalescence of the water droplets. So in this region, we have stable emulsions. And the orange zones reflects only the sedimentation of the water droplets without coalescence, leading to the appearance of an oil phase with very small amount of dispersed water drops on the top. This behavior is representative for the stable emulsions. And the stable emulsions in this case was always with surfactant concentration higher, the CMC. We also measure the average size of the water droplets, and the size of the droplets is higher for low concentrations, which favored the phase separation. As we can see in this graph, they increase very fast with time. However, for the high surfactant concentrations, the initial water droplet size is small and increases very little with time. The study of the stability of water in oil emulsions confirmed the behavior observed previously for 2 droplets. So as a brief conclusions about these parts, we could see a formation of a solid-like interface when at the droplets, when the elastic modulus rises, because of the molecular interactions, rearrangements, or possible multi-layer buildup. When the coalescence occurred, the drainage time did not present relation with the aging time, and the non-coalescence was observed with the formation of this structure at the interface with strong elastic properties. The coalescence process can be better understood by comparing directly the aging times of the interface with the characteristic times obtained by the viscoelastic characterization of the interface, rather than with the equilibrium times of the interfacial tension. And also the study of drops with of millimeter size is representative of what occurs on the scale of an emulsion composed of micrometric drops. Those results have already published in this paper. And now we are going to evaluate it with more detail, this structure formed at the droplet interface. So we've already seen that this structure will influence on the coalescence process, and we can see better this structure with this video when we increase the drop volume, and this increase promotes the break of this interface. However, due to technical limitations and the size of the droplets analyzed in this case, we could not detail the morphology of the structure. So in this part of the work, we are going to do a microscope analysis of the aging drop interface using a single small droplet. To do this, we produce the emulsions using a microfluidic device. Then we collect it, and its interface will be monitored in a microscope during a day. Here we have an example of the drop aging. In this video, the focus on the edge of the droplet, and we record the time evolution. We can see the appearance of small droplets that are emerging and moving at the interface along the time. And 3 hours later, we change the focus to the top of this large droplet, and it was possible to see that the interface was populated with small droplets. So as this process occurs without an addition of fluid and extra energy over time, it could be concluded that it happens due to spontaneous emulsification at this oil-water interface. This phenomenon has already been reported on in previous works with different kind of fluid systems and different approaches. For example, here, Gonzalez and Arauz studied a fluid system composed by deionized water and dodecan containing SPAN 80, the same surfactant used by us. And they also evaluated a water droplet interface and could see smaller droplets around 2 microns with a very narrow size distribution appearing at the water interface that increase in size with time, and also detached from the big drop going to the PUC region with time. They suggested that as the water is put in contact with the oil, a multi-layer assembly of surfactant is formed. Then this surfactant film swell and water bridges are formed, undergoing an instability transition that promotes their break in the small water droplet, reducing the internal energy. Starting our results, here we are going to compare droplets with different surfactant concentrations and same initial drop size. The first image represents the water drop at the beginning of the experiment, showing a clean interface of the drop for all of them. And we can see with time, smaller droplets are appearing and covering the spherical surface of this large droplet. And after one day, the small droplets cover the entire surface for all surfactant concentrations. However, depending on the surfactant concentration, the drop interface can take longer to be covered. For example, for the smaller concentration here, even after 3 hours of aging, the droplet interface still has clean regions without droplets. Emphasize the theory that the amount of surfactant determines the kinetics of the process. Another important observation here is about the size of the small droplets that increases with time for all surfactant concentrations analyzed. Also, when we change the focus, we can see that after the interface becomes saturated with these small droplets, they will detach it from the drop to the PUC region. And finally, we can see in these images that only for the highest surfactant concentration, the drop interface will buckle, losing their spherical shape. This probably occurs because at high surfactant concentration, the rate of self-emulsification is high, such the interface becomes saturated by the small drops relatively quickly, forming a solid-like structure at these interfaces. This analysis presented up to now was focused on the interface aging of a single droplet. However, by analyzing a group of drops, we can note the same phenomena. Here we have an emulsion of monodisperse water drops, and initially we observe that the drops are separated and the interface is clean. As time evolves, the drops move and touch each other, and the self-emulsification leads to the formation of an interface structure. But even with the contact of the neighboring drops, the coalescence was not observed. When we increase the zoom, it's more clear to see the contact, this contact region of the two droplets that forms a structure with the smaller droplets that hinders the coalescence, as already discussed before. And finally, as the last part of this work, we are going to propose a method to destabilize this structure formed at this interface. So because we observe that those emotions are very stable, has already these droplets have an interface at the surface. And for this analysis, a microfluidic device was constructed, composed by two parts. One to produce the droplets, the part A, and the other one with a constriction to remove this droplet layer that forms around the big drop. 3 analyses were performed. In the case 1, the drops were collected right after the production. So the aging time of the drops were short. After that, for the case 2, the idea was left the drops for a long time into the line before collection to provide the necessary time to the drop interface aging. And finally, in the case 3, these drops with high interface aging are passed through the constriction before collection to remove this interface. Here we have the experimental setup, and with these videos, we can see better this process. In this first one, the drops are being produced and collected with short residence time. As we can see, they are produced and right after collected. And with this second one, we can see that the droplets are formed, and before collected, they are passing through a long line, provide the necessary time to age in this interface, and then they are collected. With this image, we can see the microfluidic device with the both parts to produce, and with another one with a constriction through the droplets are passing through. And starting our results, we are going to present firstly the behavior of a single drop passing through this constriction. For this analysis, the drops were produced and kept inside the capillary for the desired aging time. With these videos, we can see the visualization of the drops produced And with the other one, we have the droplet without aging, interface aging, showing that the drop elongates as it flows through the constriction and regrinds its shape without losing mass. However, for the flow of the droplets with 1 and 2 hours of interface aging, the drops elongate as it flows through the constriction, increase the surface area, and its interface structure is disrupted. The flow leads to a strong shear stress along the interface. And downstream of the throat, we can see that the droplet interface is clean, and we have a cloud of small droplets on its trailing edge. The apparent size of the drop falls as the mass associated with these small droplets formed at the interface was removed from them. Once observed the possibility to promoting the cleaning of this interface, we tested if this phenomena could be used to destabilize water in oil emulsions. Then we produced monodisperse emulsions, and we could see that with emulsions prepared with the smallest surfactant concentration, the behavior observed was similar. In this first one, in the case one, the drops of emulsions was produced and collected with short aging time of the droplets. As we can see in this table, the residence time was equal 0.8 minutes. In this case, we can see that on the bottom of these vials, there is a free water layer resulted from the coalescence of these droplets. And in the case B, the droplets were collected with long residence time, and the emulsions remain stable over time once there is no appearance of a layer of free water on the bottom of this vial. And in the case C, these droplets with long residence time were passed through a constriction, allowing the removal of the small droplets at the interface formed with the spontaneous emulsification. And then once the interface is clean again, after being collected, it's possible to observe the formation of this free water phase on the bottom of the vial, which characterize the coalescence process. So we could separate this phase passing through the constriction, these droplets. In this case, we can also see that on the oil layer above the emulsion, this oil layer is a little opaque due to small droplets that were removed from the interface of the large drops and then remain suspended in the oil phase. However, for the highest surfactant concentration, the first case already presented stable emulsions. So even with short residence time, the droplets maintain stable on these emulsions. And even passing through dust droplets for the constriction, we could not achieve the phase separation. As a brief conclusions about the study of this structure at the interface and the way to promote the destabilization of these emotions, we could see better this morphology of this structure at the interface. This interface was formed by small droplets that appear by spontaneous emulsification. The kinetics of appearance of the small droplets on the interface depends on the surfactant concentration. And we could see that the film formed at the interface saw at the first part of this work actually was a group of small droplets at this interface that somehow interact with each other, forming a network that remains stable this interface of the large droplet. We propose a possibility of the emotion stabilization. And we could see that for low surfactant concentrations, we achieved the result expected, that was promote the separation phase. However, for the emotions produced with high surfactant concentration, they presented high stability, and even with short aging time, we could not observe the coalescence of them. As of our final remarks and suggestions for future works, in this work, span 80 concentration plays an important, we could see that plays an important role on the dynamic of the interfacial rheology and on the coalescence process. We also saw that an excess of span 80 favors the non-coalescence, as the film strength increases with the surfactant concentration. The formation of this solid-like interface was related with the strong elastic modulus, and actually was not a skin that formed this solid-like interface. However, is an agglomerated of micro droplets formed through the spontaneous emulsification that will form a network and maintain the stability of this interface. And finally, a way to destabilize these emotions was proposed, but based on the removal of this structure at the interface during the flow of the drops through a constricted capillary. For future works, we suggested change the water phase to a saline water more compatible with the water produced in the oil industry. Do a deeper investigation into the growth rate of the droplets at the interface as a function of the surfactant concentration, to obtain a time window in which it would still be possible to obtain the coalescence and consequently phase separation. And finally, improve the phase separation constructing a device to evaluate an emotion, simulating the flow in a capillary tube. This device could be a mesh formed by several capillaries with constrictions through which the emotions pass before being collected. So thank you for your attention.